Hello my dear students and welcome back to my channel. In today's video, we will be learning the topic restriction fragment length polymorphism, or which we commonly say as RFLP. This is an important topic for your class 12th biotechnology. So let us begin what we mean by this term. So first thing which we notice in this term is the word restriction. This word comes from restriction enzymes. We have learnt about this enzyme in our previous videos. So, what are these enzymes? These enzymes are known as molecular scissors. Why? Because these enzymes cut the DNA at specific sequences. So, these are endonucleases, and they recognize a specific sequence in the DNA and cut at those sequences. So these are known as molecular scissors. So the first word in this RFLP comes from these enzymes. Now the second word, fragment. So if we will cut DNA with the enzymes, then fragments of DNA will be produced. So this fragment comes from there. Then the third term, length. The length of the fragment means if a DNA is cut with these enzymes, then some fragments will be generated which will have the length more, and some fragments will be generated of shorter length. So this length comes from there, and this polymorphism term comes from the fragments and the lengths which are generated. And when we will compare these with other individuals, then the number of fragments or the length of the fragments will be different. So let us first see what happens. So if we say there is an individual A and there is an individual B, so we have in humans, we have our 99.8% similar DNA. Means my DNA is 99.8% similar to any other human. Means only 0.2% difference is there between each one of us. And we know that everyone is unique. Means I am different from any other human, or you are different from any other human. But we all have 99.8% DNA which is similar in everyone. But the difference which makes us unique is within this 0.2% difference. So what happens when we are saying that the DNA of two individuals is 0.2% different to each other? So this RFLP, when we will perform this RFLP, then what will happen, or what are the steps of RFLP? So suppose there is an individual A and there is an individual B. So first of all we will isolate DNA. First of all we will isolate DNA from both these individuals. Let us say this is the DNA isolated from A and this is the DNA isolated from B. Then the second step, we will restriction digression with restriction enzyme, and we will use the same enzyme in both these DNA. For example, if we take EcoR1 and we know that the site of EcoR1 is GAATTC. So wherever the sequence will be GAATTC, the EcoR1 will recognize that sequence and cut at that site. So when we are saying that we have 0.2% dissimilarity between the two individuals, which means that the sequence of DNA will be different in both the persons, And when the sequence of DNA is different in both the persons, so the restriction enzyme site will also be different in both the individuals. So when we will digest this DNA with the enzymes, so suppose here there are 3 sites, okay? Suppose there are 3 sites of the enzyme found in individual A, and suppose there are 4 sites found in individual B, because there is dissimilarity in the sequence. So here the enzyme could find 4 sequences and will cut at that sequence, and here the enzyme got only 3 sites to cut. So after this restriction digestion, we will run an agarose gel. Now what is an agarose gel? An agarose gel electrophoresis is performed where the DNA fragments migrate according to their length, or we can say according to their size, and the smaller fragments migrate faster and the larger fragment migrate slowly. So when we will perform agarose gel electrophoresis, then what happens if this was the DNA and there were 3 sites of cutting? So how many fragments we got? We got 1, 2, 3 and 4 fragments, and according to their size the fragments will form here. Now if in individual B there were 4 sites, so how many fragments we will get? We will get 1, 2, 3, 4 and 5, and these sequences will also get arranged according to their size. So here we got 5 fragments. So we can clearly say that there is difference in the DNA of these 2 individuals, and this fragment pattern, it can also be used as a DNA fingerprint of these individuals. So this was all about RFLP. Now we will see with an example how this works or where is the application of this technique. So the first application is in forensic science. It is used in forensic science. So suppose there is a crime site, any crime has happened and the police has found a DNA sample there. DNA sample in the form of hair or skin tissue or blood sample. So what they will do, they will isolate the DNA from that sample. सैंपल से क्या किया, DNA आइसोलेट किया और फिर क्या किया, RFLP रन कर दिया, एंड देन दे विल सी द सस्पेक्ट्स इफ दे हैव, मान लो वहाँ पर 3 सस्पेक्ट उसको मिले तो उन तीनों सस्पेक्ट्स का भी DNA आइसोलेट करेगा, तीनों सस्पेक्ट का S1, S2 और S3, उनसे भी क्या किया, DNA आइसोलेट किया और RFLP लगा दिया। अब RFLP लगाने के बाद उसने क्या किया, एग्रोस जेल में एक तरफ लोड कर दिया सैंपल, एक तरफ लोड कर दिया S1, एक तरफ S2 और एक तरफ S3। तो जो हमारा सैंपल था उसमें मान लो ये पैटर्न आया और जो हमारा सस्पेक्ट था उनमें जो पैटर्न थे वो इस तरीके के थे। तो अब यहाँ से उसने मैच किया कि जो हमारा सैंपल का फ्रैगमेंट था ये किससे मैच कर रहा है, इससे, ये वाला फ्रैगमेंट किससे मैच कर रहा है, इससे, ये फ्रैगमेंट किससे मैच कर रहा है, इससे। इसका मतलब कि जो सस्पेक्ट 2 था दैट वाज द मेन क्रिमिनल एट दैट साइट। तो यहाँ पर हमें फॉरेंसिक साइंस में भी RFLP के हेल्प से हम क्रिमिनल्स को पकड़ सकते हैं। इसी तरीके से अगर पैटर्निटी इशू है, इफ़ देयर इज़ एनी पैटर्निटी इशू, तो पैटर्निटी टेस्टिंग में भी हम RFLP को यूज़ कर सकते हैं। इन द पैटर्निटी टेस्टिंग वी कैन ऑल्सो यूज़ दिस RFLP। अब एक बात केवल और जानने वाली है हमारे लिए कि हर एक इंडिविजुअल का जो RFLP पैटर्न होगा दैट विल बी डिफरेंट एंड यूनिक, इसीलिए हम इसको कई बार DNA फिंगरप्रिंटिंग भी कह देते हैं। लेकिन केवल जो मोनोजाइगोटिक ट्विंस होंगे, जो भी मोनोजाइगोटिक ट्विंस होंगे, उनका जो RFLP पैटर्न होगा दैट विल बी सेम। सो ओनली मोनोजाइगोटिक ट्विंस will have the same RFLP pattern। बाकी जितने भी इंडिविजुअल्स हैं सबका जो RFLP पैटर्न होगा that will be different and unique, and therefore it can also be termed as a fingerprint. So this was all about the RFLP. I hope you understood this topic. If at all you have any comment, any question, please write down in the comment section below. I'll answer those queries. And if you have not subscribed to my channel till now, please do subscribe to my channel and like and share it with your friends. Thank you.